Hello everyone, my name is Sara from Ruđer Bošković Institute in Zagreb, Croatia, and today I would like to present to you the results of our study about embryo-like features in developing Bacillus subtilis biofilms. So, the relationship between phylogeny and ontogeny has been discussed in the 19th century, and the first proposed models of correlation between morphological and evolutionary development assumed their parallelism. And probably the most known is the idea of Ernst Haeckel, who claimed that the organismal development or its ontogeny recapitulates its evolutionary history or its phylogeny. Nowadays, the recapitulation models are mostly abandoned, and the majority of biologists accepted the so-called hourglass model and the phylotypic period. The hourglass model has been derived from von Bayer's law of embryology, and it assumes differences in animal embryos during early and late development, and their similarity during mid-development, namely the phylotypic period, when the basic body plan of a certain phylum is being established. During the last decade, several studies have proved the existence of the hourglass model on molecular level, and in one study from 2007, the authors used the phylostratigraphy method to determine the evolutionary age of genes. And in this method, the protein sequences of an organism of interest, in their case that was the zebrafish, are compared against protein sequences from the species phylogeny. So, if a protein from a focal species has its pair in the organism from the beginning of the phylogeny, or in the oldest organism, it will be placed in the first or oldest so-called phylostratum, and accordingly to where the protein sequences may have their pairs, they will be matched to that phylostratum. And if the protein sequence has no pair in the phylogeny, meaning it is only in our focal species, it will be placed in the highest, the youngest phylostratum. So, by combining evolutionary age of genes with gene expression levels, they then calculated the transcriptome age index, or average evolutionary age, throughout different developmental stages. So this index, TI, the higher its value, that means that in that time point are preferentially used younger genes. So in zebrafish, during early and late development, evolutionary younger genes are used, while in mid-development or in phylotypic period, evolutionary oldest genes are used. The same thing was then confirmed in plant embryonic development, but these authors went one step further, and they calculated the transcriptome divergence index. So this is a measure of divergence between a focal species. This was done in Arabidopsis thaliana, and closely related species, some other Arabidopsis. And they have found that more divergent genes are used at the beginning and at the end of development, while less divergent genes are used during mid-development. The same thing was then discovered in yet another multicellular lineage, fungi. So, although animals, plants, and fungi developed multicellularity independently, they all show the same pattern of phylogeny-ontogeny correlation. And this transferability of phylotranscriptomic tools across clades and likely universal patterns of phylogeny-ontogeny correlation in eukaryotic multicellularity prompted us to test this approach on bacterial biofilms, multicellular and most common form of bacterial existence in nature. So although bacteria are traditionally considered unicellular organisms, some of the researchers consider them multicellular because they show a lot of collective properties. So Bacillus subtilis is a frequently used model organism in bacterial biofilm research, and it shows many properties of multicellularity, like cell differentiation or division of labor, cell-cell signaling, or morphogenesis. So these properties are often seen as collective behaviors of independent cells, but alternatively one can also look at these characteristics as organizing features of a multicellular individual. So we took the latter perspective and sampled 11 time points covering a full span of biofilm ontogeny from its inoculation until 2 months of age, and recovered transcript expression values for more than 4,000 genes by RNA sequencing and almost 3,000 proteins obtained by liquid chromatography mass spectrometry. We then linked those values to evolutionary gene age estimates obtained by phylostratigraphy to calculate transcriptome age index. So if one assumes that expression patterns across biofilm ontogeny are independent of evolutionary age of genes, then the type profile should have a trend close to a flat line, or other possible scenarios that would reflect a macroevolutionary imprint would include, for example, hourglass or recapitulation pattern. So in Bacillus subtilis, we found a recapitulation pattern where early time points of biofilm growth express evolutionary older genes compared with mid and late time points that exhibit increasingly younger genes. RNA sequencing also revealed 3 distinct periods of biofilm ontogeny: early, mid, and late. We also analyzed the dataset by looking at more recent evolutionary history by estimating evolutionary divergent rates between coding sequences. So, and we found that in Bacillus subtilis and closely related Bacillus licheniformis, more divergent genes are increasingly expressed toward later time points of biofilm growth. And although we had less developmental time points for protein isolation, the recapitulation pattern was also observed at the protein level, with older and less divergent genes expressed at the beginning, and younger and more divergent genes increasingly expressed at later time points. Then, to find further parallels between biofilm growth and multicellular development, we looked for expression patterns of some of the defining features of development in complex eukaryotes, and find that Bacillus subtilis transcription regulators cumulatively have the highest transcription in mid-period biofilms, and that during this period almost all of them are transcribed above the median of their overall expression profiles. So this mimics developing transcriptomes in animals, where embryos show increased expression of transcription factors. So similarly, cell-cell signaling genes peak in transcription at 3 days, suggesting the most elaborate cell-cell communication at the time point when the biofilm gets its typical wrinkled morphology. The functional category enrichment analysis revealed a tight control where every time point expresses a specific battery of functions, and some examples include protein phosphorylation, which is an important mechanism involved in cell signaling and differentiation, both in eukaryotes and in bacteria, and it plays a critical role in biofilm formation. Phosphorylation genes enriched in mid-period biofilms likely reflect various types of cell differentiation in this growth phase. Then swarming genes, which are important for rapid colonization, peak at the onset of biofilm formation. The enrichment of genes that lack functional annotation at 2 days probably reflects the incomplete knowledge of the molecular mechanisms that govern early to mid biofilm transition, and the statistical analysis of these genes revealed that they preferentially originate from the ancestor of Bacillus subtilis strains. And this parallels development in animals where phylogenetically restricted genes are involved in the embryonic transitions and the generation of morphological diversity. So in this study, we show that phylogeny-ontogeny correlation and stage-organized gene expression architecture should be added to the list of properties that qualify Bacillus subtilis biofilm growth as a true multicellular developmental process, like those in complex eukaryotes. And multicellularity is not a rare evolutionary transition, as it has independently evolved many times in various lineages. However, it seems to be governed by the similar basic principles that include a macroevolutionary imprint. So at the end, I would like to thank everyone who participated in this study and who made all of this possible. And finally, thank you for your attention.